Okay, so welcome to System 1, Infection and Immunity module. Today I'll talk about Introduction to Protozoa. Protozoans are a single-cell eukaryotic organism with the subcellular organization. Some of these protozoans live inside of the cells, that's why we call it intracellular protozoans, and some live outside of the cell, that's why we call it extracellular protozoans. So these parasites have either direct life cycle or indirect life cycle, and some of them has a vector-borne life cycle. So protozoan parasite lead to the death of the animal due to high severity of the disease and also reduce the productivity. Some of the protozoans parasites have a zoonotic importance that include Toxoplasma, Cryptosporidium, Giardia. We'll talk about protozoan parasite is in class session. So there are four subgroup of protozoan parasite that they have veterinary importance based on their morphology as well as locomotion. We'll talk about these in the in-class session, but very briefly, rhizopods. These are the Antiamoeba is the example of these rhizopods. Ciliates type of protozoan, the Bellentidium is the example, and some are flagellates type. They have a long tail here. Trypanosoma giardia is the example of these flagellates, and sporozoans are the one who make the sporozoites inside of the cyst, and the Thaleria, Babesia, or Toxoplasma are the example of these parasite. And we'll talk about more in detail in the in class session. Arthropod-borne protozoas are responsible for the transmission of the protozoans from one host to another. That's why we called it vector. And these arthropod-borne protozoas, they are developed into the blood of the host. That's why we refer to be a hemoprotozoans. In most cases, the impact of these arthropod-borne protozoa is on the circulatory system of the host. In the case of the intracellular arthropod-borne protozoa, they have a indirect life cycle. In the case of intermediate host, the pyroplasm suck the blood. The tick suck the blood from the definitive host, and they took the pyroplasm from there and further converted into gamete. And this is the sexual reproduction, followed by the zygote formation, and finally sporozoites will be developed in the tick, and these sporozoites is released into the saliva of the tick, and then from here the tick further bite the definitive host, and the sporozoites enter into the definitive host and further converted into do the asexual reproduction in the lymphocyte first, followed by the invasion in the erythrocyte to develop the merocytes stage. So this is how the life cycle of the intracellular arthropod-borne protozoan parasite works. In the case of the intracellular protozoan with the direct life cycle, I can give you example of Amaria here. Amaria is the protozoan parasite that caused the disease called coccidiosis. We'll talk about that one in the in-class session. Is a protozoan disease in the animal. What happened? Four sporocysts will be developed within the circumplasma of the oocyst, as shown in this figure. In this picture, each contain two banana-shaped sporocytes that is right into the middle of each sporocyst. In the case of Cystoisospora, this is when other coccidian parasite cause disease in dogs and cat. Amelia normally cause disease in the cattle and sheep, but the Isocystospora cause disease in dogs and cats. They contain two sporocysts, and each contain four sporozoites in the within the oocyst. In the case of the intracellular protozoa with the indirect life cycle, we'll talk about this life cycle more in detail in the in class session. But the life cycle here the life cycle is completed within 3 to 10 days, and the animal pass millions of oocysts in their feces. So how this life cycle work? in brief. Because the indirect life cycle, the intermediate host form a asexual cycle, and the definitive host make a sexual life cycle, and in the environment, oocyst will be seen in the environment. How it works: bradyzoites, they are developed into their cyst formation develop into the tissue of the host, of the intermediate host, and from here it was eaten by the definitive host and converted into merozytes, and the merozytes further go into the sexual reproduction, form a gamete formation, followed by zygote formation, and finally unsporulated oocyst will be come out along with the feces in the environment from the definitive host and converted into sporulated oocyst, and this sporulated oocyst further eaten by the intermediate host, and they go into the blood of the intermediate host and converted into sporozoites and teichozytes, and this teichozyte further deposited into the tissue cyst, and the bradyzoites will be developed there. This is the how the brief life cycle of the indirect life cycle of the intracellular protozoas works. So finally, this lesson summary, you understand the basic biology of the protozoas, also define the basic life cycles of the protozoa that include arthropod bone life cycle, direct life cycle, or indirect life cycle. Thank you for your listening.